We want to once again welcome each and every one of you. I can see Abiodun Musludin. I can see Abisoye Dolphin. I can see Adiola Allah. I can see Aina Oluwatoyi. Please forgive me if I destroy your name. I can see Anna Sonye Giftola. I can see Chidinjoko. I can see Chukuma, Chidubem, Daniel Irene, Dr. Yinka, Akin today. I can see Elizabeth, uh, yeah, Sanyolu. I can see Emerua, Akuna. I can see Esther, Yankati. I can see Gift Abel, Harry, Paris, Harry. I can see Ibukun. I can see Isaiah. I can see Israel, Congo. So you are all welcome, Jimo. You are welcome, Azoto Gechi. A very big welcome to all of you. Ofolabi, Oluwabenda John, Osano Efe. Yes, I can see Fam, Edge, I can see Prince. So you all are welcome to this February free workshop on Chemoinformatics. So we want to say another welcome. We had same training like this first week of January, and it was awesome. That was I think most of us joined us in that training first week of January. We had Marco 4 modeling. It was so great on Marco 4 modeling. And here again we are first week of February on Chemoinformatics for life sciences. So a big congratulations again to everyone that made it this period. On behalf of Hendeb Industries Nigeria Limited, we are saying congratulations. Right? I know we are expecting a lot, and definitely we shall get a lot. This training has been put together by School of Computational Chemistry and Biology. We are one of the sub-units in charge of training in Hendeb Industries Nigeria Limited. So Hendeb is an industry that is growing with a vision of developing chemotype and prototype, and also patenting products. Yes, and that is why we are not just after training and publishing papers, but we keep saying it that we are after product development. And that is the only way we can then collaborate with the industries. So this is what we preach every day, that if those in academics now begin to prioritize product development, that is the only way the industry can then come to our institutions and collaborate with us. If not, all our research will end in bench and paper. So once again, you're welcome to Chemoinformatics for all life sciences. And I'm privileged to be your facilitator. My name is Osiago Henry Chibreze. I'll quickly share my slide with us while the training begins. So you'll be expecting a lot of things. So much will be expected, and we're going to try our best to see that we achieve what we have in this free training. So I'll quickly share my screen and see what we have. It has not been easy. We have been from one class to another since Monday. It has been from one session to another session. And some of us here has been following up in all our sessions. It's as if they say, anywhere you go, we'll follow you. Thank you for that. Okay, can you see my screen? If you can, just let me know. Can you see my screen?
Yes, yes, sir.
All right.
Yes,
So
sir.
this is why we are here. Chemoinformatics for life sciences, exploring chemical and natural product data. Then we must
Okay.
know that this training,
Hold on.
when we hear chemoinformatics, when we hear chemoinformatics, is not only for the chemist. Yeah, it is not only for the chemist. It is for everybody. Please, if your mic is on, kindly mute your mic so to avoid interference. Thank you. So it is for everybody whose discipline has something to do with chemistry. And I've really realized that there is no discipline in as much as science is concerned that does not have business with chemistry. There is no discipline. Please, if there is, you can quickly let me know. But I don't think if there is any discipline in science that if you trace little thing from there, you must have a trace in chemistry. Even those in engineering, of course, we have chemical engineering. So uh, that means everybody needs this knowledge of chemoinformatics in as much as you are working with chemicals. So is there any discipline here that you don't handle chemicals? Is there any? Your discipline, people don't handle chemicals. Oh, so all of you handle chemicals, right?
Yes, sir.
Yes, sir.
Okay, Asoka, people don't used to handle chemical.
Yes, sir. We do. Biotechnology has a lot to do with chemical
Uh-huh.
replicating agents, lysing agents, and the rest.
Yeah. Is there anyone for microbiology? Because we used to argue that they don't have business with chemistry. Microbiology med lab. Yeah. So do you have anyone for microbiology here? Okay. It seems they saw the title and they ran away. So even
Yes, sir.
microbiology even microbiology has something to do with chemistry. I remember when I was teaching my undergraduate students that we are assigned to our industry for on-site internship training. So they were for microbiology. I told them, see, I'm a chemist and you need us. They were arguing so much, microbiology. I don't know how they feed the knowledge they gave them. But I told them, okay, let's look at it this way. You want to we in microbiology we have a slogan which is microbe. All of us are all microbe. We are all microbes. What do we use to grow these microbes? We use culture medias. What are these culture medias? Chemicals. Chemicals. Some of them, most of them are chemicals used for growing of microorganisms. Now, even if you are preparing culture media, you take note of concentration. So we all have business. If the microbes have business to do with chemicals, so we are here for chemoinformatics. Once again, thank you for coming around. The training objectives, which we release, we cover the development of quantitative models. So part of what we shall be learning is how to develop quantitative models that we correlate
Your slide.
molecular structure with activity or properties. Yeah, we shall learn how to develop models that will link molecular structure, that is the structure of a chemical, that will link it with that would link it with the activity, the biological activity of that chemical, and then also link it with
in my
the
end.
property. You can see my slide.
Yeah, it is
We
now.
can see now.
All right. So we also see how we can build models that will relate our molecular structure with their properties. We'll be able to train our participants on descriptor calculation, how to calculate some chemical descriptors, build models and validation techniques. We shall have practical exercises focusing on drug, natural product, and environmental applications. Overall, the training will help support rational and predictive research. So I'll quickly go to my slide that I have for today. Hope my slide is still visible.
Yes, sir, it is.
All right. So this is chemoinformatics. So this is chemoinformatics for life sciences, chemical and natural product data curation. Just like I told our participants in the 1 month training on pharmacology, we had our 7th class yesterday, and I told them that one of the goals of this chemoinformatics free workshop is to enable participants to develop mini personal database, even if it would not be as large because it's just a workshop to expose you. Should be able to develop a mini personal database, and that will form your mini project. Because in all our training, you must deliver a project. If it is for a free workshop, you deliver a mini project. But if it's for a paid training, then you deliver a full project. That is a manuscript. So we shall quickly look at what is chemoinformatics. A lot of us already have idea of what chemoinformatics is. Just like our sister departments, which is also incorporated in our training, those from computational biology, we have bioinformatics. So bioinformatics is for biology, while chemoinformatics is for chemistry. Please, if you are a chemist in this platform now, I would encourage you that after now, find all means to know everything about chemoinformatics, because you cannot be a chemist and you know nothing about chemoinformatics. Same way, if you are into the field of biology, then make sure you grab everything you need in bioinformatics. Do we have trainings coming up on that area also? So chemoinformatics is an interdisciplinary field. It is an interdisciplinary field. That means it cuts across so many disciplines. And what it does is that it combines chemistry, computer science, mathematics, and information technology. So it's very broad. But in summary, is that aspect, is that field that uses computer to generate chemical informations. But you need little of mathematics. You need mathematics because you create models. Models are already built to mathematics. Just like you imagine someone studying computer science, you are doing programming, and you don't know mathematics. It's an error. It's very wrong. Because the models you are building, most of them are mathematical functions. Like the functions in Excel which we use are mathematical functions. So chemoinformatics is equal to chemistry plus computer plus mathematics. What are they doing? The mathematics will develop a model that we can now use the computer through the model to retrieve, to analyze chemical information. So one might ask, what kind of chemical informations? Any kind of information you need with regards to a chemical. With a chemical, you can actually use chemoinformatics to get those informations. And these informations are usually stored in a database. Just like we have the simple Microsoft DBMS system, that is Microsoft Access, and most of us know how to use it in building database for their organization. So we can also use that idea to build a database for our chemical compounds. You cannot work with a chemical without knowing the information of that chemical. When some years, I was privileged to also train some year one students in the lab in GMP, laboratory manufacturing process, and GLP, general laboratory practice. So in the GLP training, I told them that you don't use a chemical in the lab when you don't know anything about that chemical. At least you look at the hazard symbol. You look at the hazard symbol, very important. So that will even tell you how to use that chemical and the hazard associated with those chemicals. Now, how did they know about those hazards? How did they know that chloroform, when you inhale it, is toxic upon inhalation? How did they know that HCl fumes is toxic upon inhalation? It's due to the knowledge of chemoinformatics. How do we know the melting boiling point of all these chemical substances? Is from the knowledge of chemoinformatics. So now some of this knowledge are gotten from laboratory experiments, lab data, lab experiments that were generated in the lab, and they were able to develop those data. While some of them are generated through computer models and algorithms. So you can set up a model that will use other parameters to generate values for similar parameter. So you must have computer knowledge combined with small mathematical algorithm, then with full knowledge of chemistry. Now, it plays a critical role in drug discovery. You know, drug is chemical. This week, I was interacting with some students in SS1 in Nigeria school. So I told them to define food. Okay, I defined food for them as a chemical substance. I started with that food is a chemical substance, and they were shouting, food are chemicals. And you cannot, for those who are in food science, food tech, you need knowledge of chemoinformatics. Even drug, of course, drugs are purely chemicals. Drugs are chemicals. So how can you produce a drug? How can you develop a drug without knowledge of chemoinformatics? You are heading to nothing. Because it is chemoinformatics that will provide all necessary information that you need about that chemical, about that drug. Not only in drug discovery, in material science. I received, just this week, we received, in our lab, we received 2 jobs. Coincidentally, these 2 jobs are PhD, two PhD students. One of them is in Federal, I think that should be in Futo, Federal University of Technology, Owerri. One came early this week, one came today. But I'm interested in the one of today because both of them are actually doing anti-corrosion, and they are applying computational tools in anti-corrosion. They are using, like the one of today is using DES DS-based nanocomposites in fighting corrosion of mild steel. So now you need knowledge of chemoinformatics to be able to retrieve information on the chemical behavior of that material, because you want to build an anti-corrosive and anti-corrosion agent that can be used to inhibit corrosion of mild steel. So that is from material science. That is from physical chemistry and material science. So you can apply this knowledge of chemoinformatics in that area to build materials that you can use to fight against corrosion. So not only material science, we apply it in environmental chemistry. Let's say you want to develop a substance that can help in bioaccumulation as a biocoagulant to coagulate wastewater, public cassava meal wastewater, which is a common waste. You need the knowledge of chemoinformatics so you can be able to know the properties of that your coagulant which you are using. Okay, let's say you have a case of heavy metal pollution in wastewater, and then you want to do a study that will absorb this heavy metal. That is adsorption. You can develop an absorbent with knowledge of QSPR, that is quantitative structure property relationship. You can develop some chemical substances with the right chemical property that will fit in properly as an absorbent, depending on the type of absorption you want, if it's a chemosorption or a physiosorption. And then you must assess the absorption parameters like effect of contact time, effect of temperature, effect of absorbent dose, pH, and all the rest. Then you can even go as far as assessing the thermodynamic stability of that absorption. You can go as far as generating parameters on the ISO times, the ISO times of that absorption. You can look at the Fendlish, the Henry isotherm. You can look at the Landmore isotherm and others as much as possible. So all this data are what we need in chemoinformatics. Without you going to the lab, you can tell if this substance can be useful in this area without you going to the lab. This you can get from knowledge of from chemoinformatics. It's an interesting area. Is it in the area of the agrochemistry? In my team, we have done a lot of work on agrochemistry, biopesticide, insecticide. We have done a lot of work on that area. You need that's an insect that a chemical has the ability of controlling some pests. That means it's as a result of the chemical property. So you can generate. Please mute your mic. Okay, thank you. So you can generate chemical informations that can make that substance to be used as a biopesticide, as a botanical pesticide. So this is not only for those in chemistry. All field of study can imbibe this knowledge and apply it. Unfortunately, it's not being taught in our institutions. Now, the overview of chemoinformatics. One of them is data storage. I'm going to dwell more on this part because that will form our first hands-on. So once I'm done with the slide, we shall now look at hands-on, brief hands-on for today, while tomorrow is totally hands-on for 2 hours. Data storage. Sorry if part of what I will say now will affect some of us, but that is why we are here. How many of our departments? How many departments? I thank God we have most people here from UNIO, because when we look at the dashboard on registration, we saw a lot of participants from University of Uyo. So those universities represented here, thus our research directorate, the RD and I, research development and innovation directorate, do they have a team that help store all the data we generate through final year projects, master's projects, and PhD projects every year? Because you cannot talk about chemoinformatics without data. The beauty of science, but the beauty of computational science, computational chemistry, or computational biology is data. That is why even those in the bioinformatics, thank God for the African Genome Project. I think we need one for Nigeria Genome Project. Thank God for African Genome Project, because most of the whole genomics data we have are those in Europe, abroad, US. We lack African genetic data. But I think that call is on already on the African Genome Project. Thank God Ebenezer is actively on that aspect, alongside with Professor Happy of Redeemer University. So a lot of work is ongoing on African Genome Project. How many work is ongoing on our chemical data? On our chemical data. Of course, we will not say African chemical data, but the one we are generating in our department. Do our department store our data? I was interacting with final student today from a school nearby to our organization. So they came around and I was asking them, what is the essence of the project we do, the research we do? We are in Africa, we are after doing a research just for the score, just for the mark. That is why you can go online, pick any result and convince your supervisor, convince external examiner, and they give you your A. We need data. We need data. Let's create our data. Like I said, one of the plan is for us to build a mini personalized database. So we need data. I strongly believe that there is no final year project research that does not generate data. Even those who are working with questionnaire, the empirical studies, those who are working with questionnaire, mostly those in the School of Business, even the arts, those under arts, they still generate data. Some of us that do survey, we generate data. What are we doing with those data? Are we storing those data or we are wasting it? Is a call to everyone. Probably I may have some VC represented here, but I believe we have professors here. So they should take back to their institution. We have a head of department here. So take it back to your department. In every final year student project, masters and PhD, data are generated. Where does it end? It ends in library. It ends in distinction. In grade. So we need data. We need those experiments that we have been conducting. Let's say, for instance, now, like those in chemistry, we have done a lot of work on heavy metal concentration, maybe of water samples, probably a particular river. A lot of work has been done on that heavy metals in your department. Do you have that data? Do you create those data? Because we can use those data to do future trend. We can use those data to develop policy. We can use those data. We can now develop a database that we can use those data to correlate with other chemical information that we need. but we end up in, most of us even end up at publication. Do you know how many data we have released in the name of public? Just think of it. Somebody will beg you to publish a work with them because most of you are tired of the mails you receive from different publication companies begging you to publish work with them. So they will beg you to publish work with them. You will spend your money, do your hard-earned research, send your data, because what you are sending to them is data. You think you are any metrics. You think you are any metric. You are sending your data to them. Then they will ask you to pay them. You have to pay them for your own data. Now they have access to data, so you have to pay them to now publish your work. Work where I use my hand do with my own money. Now I still they pay you too. So you just understand that. So now we are pushing out our data. And if we tell you now to publish in local journal, it will say no, because you need international recognition. We are after international publications. We don't want to build our own country. That's why our researches are not reflecting in our local communities. So you take your data out. You take your data out. They use your data to build database. And when they build that database, you don't have access to it. They can give you institution access by collaborating with your school. Or they will not ask you to pay to have access, to subscribe to those data. Those data, you generated them by yourself. So we should start learning that data storage. So that is the first thing. That is the first thing when we come to chemoinformatics. We cannot talk about chemoinformatics when we don't have data, when there is no data. I was doing a training last, was it October? We had a training on molecular biology. Yes, we had a training on molecular biology. And during the course of that training, I introduced QSAR, that is a QSAR. Now, we noticed that one of the participants wanted to work on a lead, one derivative. So she went to some of this database and she could not see useful data. There was no much data. But this is a work that most of us have been doing in the lab, and we have been wasting it. So we don't learn how to create our data. Both the natural product data and the chemical data we generate in the lab, we learn how to create them. So first thing is data storage. So when you're able to get these data, so I hope we are following, right?
Yes, sir.
All right, thank you. It's somehow different from our usual trading, but I had to just sound it so raw. It's a bit visual from our normal training, but I think we are getting something. Now, when you have those data, so you are going back now to your HOD's office now, because most of them are there. They are in your student library. So go there, pack those data, begin to sort them, arrange them. So we'll begin to do electronic storage. Now, when you have those data, the next thing you do is to index those data. And these are what they do in all these chemoinformatics databases. You must have an index. Index means like in PubChem, if you download a structure, it comes with CID for compounds and SID for substance. So we have compound ID, we have substance ID, SID, then CID. So, and that CID comes with a number. Like all the students, we are indexed, we have reg number. we have matrix number. That's our index. So when we search, if we search with your matrix number in your database, we can generate all the information of that student. So the same way you that want to store data, you work with index. You can say Hendeb 1 or Hendeb 001, and that Hendeb 001 is for so chemical. So once you search Hendeb 001, then every information of that chemical will appear. Then you can link the database with other databases also to generate more data, more informations. So there must be an index. There must be an index. Then the other thing about database is for search. There must be a space for search, because you have generated this data and then you are storing that data with an index, so you can easily search. So there must be a search platform, search button where you can search with the name of that compound, or you search with the index of that compound, and then necessary informations will be generated from there. You just see their information, like we do in PubChem. You just type the name of your compound and you see everything you have about that compound. There is a search button, so it is related. It is interconnected. Then another thing is retriever. You must now create an avenue for users to retrieve this data. Maybe they can retrieve that data in different formats depending on the data. If they want to retrieve structural data, so you make sure that they can retrieve the structural data in the structural format, maybe as mode formats, SDF formats, as sketch format, draw format, and other format, faster format, other format that is available. You can make sure that they can retrieve that structural data. Now, if the information they want to retrieve is not a structure, maybe they want to retrieve those properties of the structure, then you create that avenue that they can now download the properties generated either as a CSV file that can be opened in ChemDraw, or they can download as PDF. So such avenue is usually created in databases for retrieval. So as this training is going on, your mindset will be now beginning to open up for how you can store your data. So just keep your questions, just be writing them down, because we are going to interact so much. Then there should be availability for analysis of chemical data. So that is still part of data storage. So you can analyze your chemical data. You can find out more. That is the analysis. You can find out more information about your chemical data. So when you click on one data, you can take it to another information that you need. And then, okay, let's see the key areas. We just finished the overview. These are what you expect in a simple database. So in the project that we shall be doing, if willing, these are what we should expect in that simple database. There are key areas in every chemoinformatics database. There are key areas. One of that area is the molecular representation. So your molecules must be represented either in 2D or 3D structures, because the molecules must be there. So they must be represented either in 2D or 3D structures. Of course, you will draw them. You can now draw them using some drawing tools like Marvin Sketch, Came Draw, Came Sketch, and other tools that you can use to draw. IC's Draw. IC's Draw too can give you that opportunity of drawing. So you must represent them as 2D and, but most importantly, as 3D. As 3D. Then you must represent them, those molecules, in a file format, like I've said before. You can generate their SMILES, which is a simplified molecular input line entry system. You can generate their, most importantly, SMILE. I love generating SMILES because it makes me smile. So you generate their SMILES. SMILE is very important. Some of these algorithms in some database work with SMILES to generate their SMILES. And you can generate SMILES with ChemDraw or ChemSketch. Generate the SMILES, store it. Generate the entry code, their mode, keep them as those structure as mode file or as SDF file, as structural data file. So these are what you need. Then you still present your structures using descriptors. These descriptors are features that have been turned into numbers. That is chemical descriptors. So there must be chemical descriptors. You can present them with their chemical descriptors, which are features that we are turned into numbers. So numerical representations of chemical features. Like take for instance, okay, those who have done food analysis. Yeah, those who have done food analysis. You do descriptor, but you may not know. If you're doing food analysis, mostly sensory evaluation. Yeah, the organoleptic properties. If you're doing sensory, you see that you use a scale of about 0 to 9. Now you have taste, aroma, mouthfeel, and others. So you convert those features to numbers. So when you test the food, you have a range. So if it tests so much and satisfies you on that range, you give a number to that range. That is descriptor. So chemical descriptors are numerical representations of chemical features. So I can turn the molar weights, the weight numbers. The solubility can now be expressed as a number. The hydrogen bond donor, so I can count how many hydrogen bond donors are in that structure, keep it as number. I can count how many hydrogen bond acceptors are there, keep it as number. I can count. So these are the features of that compound. I can count the number of rings, number of benzene rings, aromatic rings in that compound, count it, keep it as number. So I'm turning benzene ring as a feature to numerical feature in number. So all these forms, I can count the number of oxygen, keep it as number. So all these forms, the chemical descriptors, they will be part of your database. So one of the area is molecular representation. So you must learn how to represent your molecules. Then another area is the database management, the DBMS. So you must create a system that enables you to manage that database. And managing means storing and managing large chemical libraries. We have some of those database management systems like the PubChem, the KMBL, the Zinc. So where you can leverage on that. Outside representing your molecules, that system must be able to be managed properly, that people can store, people can even upload. Yeah, you can create room, people can upload their own data. People can upload their own data, and you can even accommodate large thousands of information. Do you know how many informations are in PubChem or KMBL? So you must be able to manage those data. And then chemical structure search, like exact match, substructures, similarity search. So there could be an avenue that you can search from your large data you have created. You can search Probably can even be specific compounds that has amino group. So once you type amino group, you can be able to search similar structures that has amino group and bring them up together for you. Now, another aspect of another key area of chemoinformatics is the molecular modeling. Here you can predict molecular geometry and energy using force field or quantum mechanics. So you can model your molecule. Although this aspect is rare in some of our database, in most of our chemoinformatics database, this aspect is rare. But what it does is that it gives you that privilege of predicting molecular geometry, the spatial arrangement, the electronic strength, the steric effect. You take note of those things using some force fields, like universal force fields, quantum mechanics, MM2, and others. And then some of them have space privilege for you to do docking, homology modeling, but it's very rare to see a mathematics database that can enable you do docking, do homology modeling all at the same time. So they create special database for them for proper modeling. So another area in mathematics is the QSA and the QSPR modeling. So you can use information there for quantitative structure activity relationship and structure property relationship. So this just formed our first objective, which is how we can relate structure molecules with their activity. How you can relate molecules with their activity. And how you relate by after this, but on the 18th, around 18th, yes, after next week, upper week, is the third week of February, we have a special workshop for QSAR and QSPR. Yeah, we have a special workshop for that, for QSAR and QSPR. So you can use the structures of a compound to generate the activity, even without conducting any lab work on that activity. It can also use the structure of a compound to generate the properties without doing any evaluation of physiochemical properties. So you can do that. That's one of the areas in chemoinformatics. Then we have the virtual screening. So you can use this chemoinformatics to screen large compounds. You can use it. You can fill in like the ADMED tools that is used for PK for macro kinetics. You can see that you can put in up to 1,000 compounds, 1,000 smiles, and it will screen those 1,000 smiles, generate their absorption, their distribution, metabolism, excretion, and toxicity values or properties. That is the virtual screening. So you can screen large molecules for potential lead at the same time. Now you can also use it for ligand-based studies. This ligand-based is the backbone of QSAR and QSPR. So whenever you are starting, whenever you hear ligand-based studies, ligand-based design, ligand-based docking, ligand-based drug design, what you use in ligand-based is purely QSAR, 3D QSAR, and QSPR. Now, what are the possible applications of chemoinformatics? So, if we begin to create databases, what are the applications of those chemoinformatics? What are the applications? So we can say that one of the applications of chemoinformatics here is hit and lead identification. You know, those in drug design, they are very common with hit and lead. But thank God for the vision we have in Hendeb. We have chosen not to focus on drug. So now a heat, let me use a heat is a potential compound gotten from libraries of compound. Maybe I want to screen compounds that are good as absorbents. So I have okay, let's say compounds that can be used for the removal of dye, for the ST as absorbent for removal of dye. So I have 200 compounds. If I screen these 200 compounds, so with knowledge of chemoinformatics, you can screen those 200 compounds based on the properties needed for good absorption or good removal percentage of dye. So you can screen them to 5. So those 5 now are potentials. They are potential compounds. So they are heat. So now those 5 now can further be optimized to lead. So we call it lead optimization. So you can improve the properties of those compounds by modifying the structure, adding some groups, some substituents to it that might even improve that desirable property. So we can use chemoinformatics to also do that. You can use their softwares or you can use their database to do that. So you can improve it. What you're doing is that you are converting those your heat to lead. It's called lead optimization. So you can apply it both in drug. The last training we did January, which is last week, on the beginner's course on chemoinformatics. Sorry, on computational chemistry. The project they are currently doing now is that they are building 6,000. They are building 6,000 analogs from different heats, so which we can use them for lead optimization. So you can imagine those participants in our last week training, the assignment, their project is that they are using ChemDraw to model in 3D 6,000 compounds. Some of you will be shouting Jesus. So that's one of the beauty of chemoinformatics. You can apply it in 3SAR studies, which we have already explained. You can apply it in 3SPR evaluation. We have already explained that, and we are keeping 3SAR especially for next 2 weeks. We'll take our time to explain that. So you are free to do that. Now we apply it in hazmat prediction. So if you look at a structure, you can now tell the potentials of that compound to be absorbed in the body, the potentials of that compound to be distributed in the body, to be metabolized, to be excreted, and the possible toxicities. You can do that without going to the lab. Those who are in the environment, you can check if these molecules you are using for environmental pollution control, if they are eco-friendly. So you must not go to the lab for you to know if they are eco-friendly. You can use your chemoinformatics tools to generate environmental parameters that will tell you, this is your compound, it can accumulate on plant tissues for long. This your compound is not degradable, is not biodegradable, and so on and so forth. So you can use that. You can use that for toxicity modeling, which I've explained now. So there are a lot of applications of chemoinformatics. So a quick one, there are some data source. There are some common chemoinformatics available to us now. We have the PubChem. These are common chemoinformatics too. So I believe that all of us have used PubChem before, right? I don't think if there's anyone at this point in time who have not used PubChem. Yeah. So these are simple database sources. The 10BL, they are simple database sources. The Zinc database. There are thousands of chemoinformatics database. Yeah, there are thousands of chemoinformatics database. The drug bank is also a chemoinformatics database. We have the binding DB, the Swiss Admer, the TOSNet. So there are a lot of chemoinformatics databases, but yet they are not enough. We need our own local database. Our own local database. Let me not say something. I want to say something about, because we have some foreign participants here. So I wanted to say something about Nigeria, but due to the presence of some foreign participants, we just have to monocast our country. So I will keep it safe until when we have just only Nigerians among us. Then I'm going to say that, because it's a problem in Nigeria. I don't think if it's a problem in other countries, but it's a problem in our Nigeria. So we have some challenges in chemoinformatics. Challenges like quality and standardization of data. How are we sure that the data we are using are quality data and they are standard? See, will you just use a data that one student just duped data to play with the mindset of their supervisor, and then you are going to use those data to build a database, and you want to publish those database, or you want to use them for a research work? See what will happen to you when you do that. When you use them in computational studies, they will tell you, wow, from the result, these data are good. In fact, if you look at the activity or the property, is giving you close to 80%. By the time you now go to the lab to validate what you have done, that is when you confirm that this data that we have published is not correct. One of my students, or not my students, one of my participants from Federal Polytechnic, Owana, Afikpo, at the Ebonyi State, she noticed that some of the reports, okay, she was doing some physiochemical analysis of some chemical compounds. So she generated data from Admet Lab 3.0, from Admet Lab, and generated that same data from Swiss Admet. And she noticed that they were not correlating. There were some discrepancies in those data, where for a property, Swiss Admet will be quoting like 10, while Admet Lab will be quoting like 5 or 6. You can see that's far difference. So that is a challenge we have, because for we to rely on this data, one of us doing our training, that was on Saturday, when we are concluding our beginner's course on Saturday, just this past Saturday. One of our participants shared his experience where some of these lecturers, supervisors will tell you, you are giving us computer data. You went to computer and generated a result, and you want to use that result. Most times they are not wrong, because you must ascertain the quality of the data, because you want to rely on the output you get from this data. So of what is their quality? What is the quality of that data? Was the experiment carried out at standard conditions? Some will tell us that in Nigeria, in Africa, we cannot do a work at standard conditions. So what are we doing? Trial and error. So that is a big challenge in chemoinformatics, because for us to rely on the output, on the prediction, then the data we are using should be of high quality. Now, one challenge is some of these database also are unable to handle large data sets. That is big data. So we find ourselves limited. Like yesterday, during the training, I could remember Professor Owolabi to me. Maybe he might be here also. He reminded me of a question he asked about a database that can give us information on reproductive toxicity, which ADMET Lab, Swiss ADMET, and some of these common chemoinformatics database doesn't have that data for reproductive toxicity. So these are challenges you have with data. We don't handle big data. And even if we handle big data, are they available? So for we not to have information on reproductive toxicity, it simply means that maybe they are unable to access large data on that aspect. And that is a challenge. And not that those data, they are not there. They are there, but do you know where they are? They are in the library. They are in the library. We have beautified our library with project and research work. God will help us. A challenge we have is accurate 3D conformal generations. Most of us, because of the softwares we are using to generate these structures of the chemical substance, we fail to generate their conformers, especially in 3D. Yeah, especially in 3D conformers. So that's a challenge. The integration of genomics and system biology with chemoinformatics has been a challenge. So we need a system that, thank God for Swiss. The Swiss are doing a lot of work. I think we have Swiss ADME, we have Swiss Doc, we have Swiss Biosystem, we have Swiss Sidechain. So the Swiss company, they are trying to, if we have Swiss target prediction. So they have been trying to do this multi-omics, integrating some genomics data with some chemoinformatics data. So there's a linkage. So that is a challenge because that gap is still there. If not, somebody who is studying bioinformatics should also study chemoinformatics for effective understanding. And at the same time, you studied chemoinformatics should also have a good knowledge of bioinformatics for the application. Another challenge we have is the interpretation of black box models from machine learning. Sorry. Now, when you develop a model, like machine learning uses about 2 or 3 layers, while deep learning uses multi-layers. So when you develop a model in machine learning, how do you know that that model is effective? All we're after is the outcome of the model. That model might be overfitting. That model might be underfitting. But we just use them like that. Because some of these database we're exploring, they are built with machine learning and they created a model. So do we understand the black box? That is the algorithm in that model that is giving us the output. How valid is that model? These are what we consider, because in chemoinformatics, you must build a model. You must create a model in chemoinformatics. But we have future and we have a hope. We have a hope in chemoinformatics. And one of our hope in chemoinformatics, informatics is integration with AI and deep learning for better prediction and design. So we can see how we can now bring AI-driven tools in chemoinformatics, then bring in deep learning. Because most of us, if you look at what is popular is now is machine learning. A time will come, we'll start to Talk about deep learning. The only reason why some of us doesn't have access to this is the cost of learning these next generation research tools, and even the tools, the platform to be used, you have to access them. Most of them are not free, they're not open access. Then they require programming. You can imagine telling a chemist to go and learn programming. He will tell you that one is for those in computer science. Trying to encourage a professor in microbiology to go and learn programming. He will tell you that that one is for those in microbiology. One of my friends in microbiology, she will tell you, Henry, please, once it's time for all those on our chemical work, I will push it to you. You will do it. Because it's a challenge to most people now. You have to have knowledge of AI. AI is not chat GPT. Is not Creebus or the DeepSeek we use. No, that is not. All those ones are browser AI. We have AI-driven application tools or softwares that I can use. So that is the future we're talking about. And then we have cloud computing. We have promised, we promised us this year that we are going to do a training on cloud computing. The reason for that was there was a challenge for MD, molecular dynamics, which personally I have changed our system up to 4 times because of MD, and I just decided that in our institution for now, we may not be running MD, but we have people whom we collaborate with. Mostly, we have partners in Pakistan and India, where we partner for MD simulations. But then we can do cloud computing that you don't need your data storage, your system speed, and others. You just buy space. You buy the space and do your computing. So that is where we are looking at. That is where we are looking at. Then expansion into green chemistry. We have been talking about this green chemistry. Yeah, green chemistry is for all. Is something that I will encourage every one of you to go into. And we are having a training on green chemistry starting next week. Next week, February 10th. Next week. That training has been power packed for green chemistry. I will share the flyer with us. There's a power packed for green chemistry. We are, I will not say we are the first people, but at least we are among the early career scientists who are applying computational chemistry tools in green chemistry. And if you look at the cyberspace, you barely find a work on in silico that has to do with green chemistry. Somebody had to cut my DM today, and the person is doing okay. I shared it with us on the corrosion, and the person is using DES based nano composite. And I was telling the person that what you are doing now she wants to bring in computational chemistry. I begin to share the idea that what you are doing now is doing chemistry, and is novel. There is a gap in literature. So that is the future where we can now apply chemoinformatics in green chemistry. We can now apply it in personalized medicine. That is one drug for one person. Not the one who will go to the market to our pharmacy and then we say, I need malaria drug. They will give us that same Leonard, the one they gave to the other 10 persons that came that day for malaria. They will still give you the Leonard, and then you take Leonard and you complain that this drug is not working. You don't know that the parasite in you, your Plasmodium falciparum has developed resistance to Leonard, to artemether, albefatin, and others, and we keep taking them. So we are now preaching precision medicine. That is personalized medicine. I don't think if Africa can do this. I don't know, but I think South Africa, they are ahead when it comes to genomic studies and precision medicine. So they're really ahead, and I think some part of them might be doing that, because if you look at they have in Kwaba, and so there should be a, a lot of Sangha machines there for them to conduct personalized medicine. And then we need that, we can apply that knowledge now in synthetic biology. So that is what we are hoping, that we are hoping that we can now apply our mathematics in synthetic biology. So at this point, I want to take your questions. Yeah, I want to take your questions. Then we'll do a little hands-on to introduce us to tomorrow's hands-on training. So we are going to take. So let me stop sharing my screen so I can attend to some of your questions. So let's discuss. All right, the floor is open now. Thank you. Thank you so much. Okay. Remember, we have a lesson to go back to our chefs, convert those projects we have there to data. It's a big word, but we'll do that. Okay, let me take sorry if Chinonso, I think that is an Igbo name. Okay, go ahead. You have the mic, Chinonso.
For what? No, I don't have anything to say.
You don't have a question?
I don't have question.
Because I saw your hand up. That is why.
Okay, sorry. It's a
Oh,
mistake.
are you not raising your hand up there? Yeah, you know, are you not raising is that not up there where you are?
It's a mistake. It's a mistake.
Okay. Okay. And if you just took your hand off. All right. Yeah. Yes. We expect some questions so we can interact. Okay. I will take my Professor Adiola Allah.
Good evening, sir. Good evening, everybody.
Yeah, really, Prof.
Yeah, good evening.
Okay.
My question is this.
Okay.
There's an impressive list of areas where computational chemistry is very useful. I would
Can
like
be
to know
applied.
from you with your experience that which if one is to identify a research gap, because there are quite a number of areas, which area do you think one should explore in all these areas that is really very important for our climb and for this time that we're for important for our country, like third world. Are we third world? I'm not even sure again. And what we have in our country and this time, that this particular period. So where do you think we can focus?
Okay. I want to understand that question in two ways. Do you mean, firstly, area where you can focus your computational knowledge skill on, or areas of computational chemistry where you want to focus more on?
No, I want to know. Research should not just be for the sake of research. it should be out of need.
Good.
It should be out of need, not just
Yeah,
because we want
yeah.
to do something. So I'm asking, which of these areas has a gap? That means either there's no knowledge or there's a need that the
Okay,
company will benefit from if we do the
okay,
research.
okay. Uh as one in this area, I will tell you that all the field in science needs this has a gap because we are yet to apply computational chemistry in all this field. Now, no area is negligible. Yeah, no area is actually negligible because if you look at all these areas we know in research, we are still using our traditional method, and that has hindered the progress in those areas. So
Okay.
if we cannot bring in computational chemistry to any of these areas, then you see that we'll speed up our progress. Now, but if I want to suggest, I will tell you to focus on what it depends on your discipline now. Yes, because the area to focus depend on your discipline is now discipline based. But I'll tell you that focus on environment.
Okay. Okay.
US are spending money on environment. And people
Okay.
some countries are spending money on the environment. So a lot of research. It was recently, just last year, that I just thought one of my lecturer friend, she's now in Brazil. He said, Henrio, he said, Henko, focus on environment, biotechnology,
Okay.
biotech. That area, people don't do much on that area. So I did a work with her before she traveled to Brazil. We are able to use computational tools to see how we can generate biofuel, bioethanol, using cow dung, cow shit, cow dung. We generated a biofuel from cow dung.
Okay.
Did that work with computational tools. And when we went back to the lab for validation, the result was awesome. The only challenge is that the quantity of the biofuel we got was so small. But I think we are able to do that laboratory work. So it's just for us to now do a pilot scale to scale it up. And when she left Nigeria, of course, that was not my vision. That was her vision. We just collaborated. The work ended like the work just ended like that. Whenever she will come back. So there is space for us, enough space in
Okay.
the environmental. Yeah.
All right, sir. Thank you.
Then other professions too still have loopholes, but you see the environment. Look at our environment now. Are you happy the way the environment is? Just take a stroll out of your apartment, your house. Look at the street. See how littered. You see dustbins everywhere. Can't we do something? I told someone that we are not healthy. In as much as the air we breathe is not clean. All of us are sick. All of us here, we are all sick, depending on your country. But you see those in Nigeria, everybody sick in Nigeria because the air we breathe is not clean. We can achieve clean air. That is part of what we are doing chemistry by next week, 10th of February, doing chemistry. That is what we are preaching in that green chemistry. So thank you very much for that wonderful question.
Thank you, sir.
All right. I saw one of my friends, Dr. Chidio, in Joko. Yeah, his hand was up.
Yeah, Mr. Henry, thank you very much for that wonderful presentation. In fact, I'm wowed by, the level of knowledge you have really enunciated. Thank you very much for the presentation.
Thank you.
Mhm. So
Thank you.
I have this question. For those of us in natural products that are researching in the areas of medicinal chemistry and all that, what are, the most lucrative areas that this chemoinformatics can be applied? What are possible, areas that can be applied? And how can we also design, very wonderful topics from, that, synergy between chemoinformatics and the natural products?
Okay.
Thank you.
I'm going to show you something. I'll really share my screen now. Then I will show you something. This is part of what I never wanted to say, but I think the question is here. So Nigeria does not have a phytochemical database. See, Nigeria, we don't have a phytochemical database. I don't know the people that did this work. Can you see my screen? I'll show you
No,
something
no,
because
no.
okay, it's coming up. So
Okay, yeah, I can see the screen.
all right. Now, when we get to phytoinformatics, I think we are among, yes, we are still among the earlier scientists to dive into that phytoinformatics. There is an area of chemoinformatics called phytoinformatics, and we did that during our 1 month training on macrognosy. So look at this database, Nigeria phytochemical database. I came across this database last year. Now, this database was built by some researchers in Nigeria, but it was so dry. These guys, we are giving money. It was a third fund project. So they initiated a research in third fund NRF. You know, NRF will target at least all of us put our budget between 30 to 45 million naira. It's something, it will solve some problem. So some people put up, these guys put up that proposal for creating a chemoinformatics database for phytochemicals in Nigeria. It's still loading. I would have showed you. So for phytochemicals in Nigeria. Now you can see that is a unique research area. Creating database first is a research area on its own. America has a database that was created. It was sponsored by the government. India has their own database, Impact, sponsored by the government. China have their own database, sponsored by the government. I don't know, Esther, does Kenya have a database for phytochemicals?
Not yet. I don't think there's any country in Africa
Okay, but
Dr. Henry, Dr. Henry,
yeah,
when you say database,
Prof, I can hear you. Yes.
Yeah, when you say database, is it any database? Because I know I know
The
of
network is good. I can hear you clear.
I said when you said database, I'm not sure exactly what you mean because I know I've seen like 2 databases for Nigerian plants. The first one, in fact, they're in volume 1 and volume 2 by Professor Odugbe.
Okay, can you come again?
I said when you say database, I'm not really sure what you mean.
Yeah.
Because I've seen two types of databases that I've seen. And the first one is even in 2 volumes. It's in 2 volumes. I think Nigerian plants or something. In fact, I've seen like 3. Nigerian plants or something. That one is volume 1 and volume 2. The author is Professor Odugbemi. He used to be the VC, I think, of Unilag at some point. But he's a microbiologist, but he went into this natural medicine. No, let me say plant medicine. So he has 2 volumes of work that I've seen, that I saw. Then another one, I can't remember the name. I can't remember the name. I've seen about 2. Then I saw another one.
Okay.
I saw another one in IIT. Something plants of West Africa. And they are really, it's not that they are very full, but they are not dry. That's why I'm asking that, what kind of database? Must it have like CID number and things like that? Because they just have the plant and some chemical properties and some other things like that.
Yes. Now that is our challenge. Our challenge is the ability to create data. Now, I've also come across those tests. There are some tests that I've come across on natural products in Nigeria and Africa. But are they available online?
I saw hard copies. I haven't even. I saw the hard copies.
So,
I didn't. I'm not sure I checked online, but I saw the hard copies.
So hard copy is not database.
Okay.
That is a documentary.
Hey, that's what I was asking. What is a database? Okay.
Yes. Yeah, that is a documentary that was made. But they can convert. You see, if they're not careful, that documentary, if the author dies, what happens? People might not even take it up. It will just end there most times. Because we have heard about some textbook those days that we are so good. But go to the libraries now to check about them. You won't see them again. Those details, informations went off like that. So we are saying we need a database that we can publish online, and we can use them we can also access them, we can index them, we can retrieve data from there. So this one now, yeah, Prof.
How do we verify before? Let's even say the authors have soft copies. Before uploading, how do we verify the information in these documentaries?
You see, that was the problem we said, the challenge we have in quality of data. But whoever that is getting data to document should ensure that what is documenting is valid now. So that is a problem to us in validation, because except we collect those data ourselves and build the database ourselves, that is when we can claim 100% validation of our data. But if we are now leveraging on documentaries that are available, we know what is happening in research in Africa. So we begin to wonder to what extent are those results valid.
Sorry, my network went off. I'm back.
Okay. So what I was saying is that to validate those data, the documentary, is a problem to us because it was not self-generated by us. It was generated by people who documented it. So we don't know to what extent are those data valid. And the same thing we have in our project research work. We have people who just dupe results and conclude in their work, and then we want to rely on those results to build our own database. So it now is a call for all of us who are into the academic system to scrutinize our research students, the research what they are doing. I personally, if I give you a research work, I will follow you to the lab where you're doing it. I will try my best to be there, see what you're doing. If possible, I know it's not to an extent you can't do that, but you just check what they are doing. If not, these people, once they are not there, should our results they fine. Anything they give you is the result of their finding. They will even cook it, use stamp from God. So this is a challenge we're having. The research ethics.
It's true.
Yes.
Hello, Dr. Henry. I have some contribution I want to make.
Okay, sir.
Yeah, talking about databases. Well, you've rightly said that books are not databases. Why books are not databases is that for a database, I mean, from any field, there are certain attributes that what's it called, database must satisfy before it becomes a database. And what is this? Number one, reliable storage, right? Easy access to easy retrieval. Those are those are very, very important. And again,
very important, yes.
Apart from being reliable, it must also be well secured so that nobody can just go inside and fiddle with it. Now, there are many types of databases. So a textbook can never qualify as a database. As you said, that was just a collection of literature. And again, the quantity of data of information there will be very limited. Now, when we are talking about databases, there are many types of databases. We have non-curated databases where anybody can just go there and upload something. Of course, the validity of such database or their reliability is not going to be very high. And those databases will put up a caveat there that this is a non-curated database. So that when you are going to be using it, you are going to be using it with caution. There are other databases that are curated. This is just like a library, right? Where anything that is going to that library, we have what we call curators, people that are doing quality control of the kind of data. So you can't, not just anybody can go there and upload, right? That is the next level of validity in terms of the source of the information. There are also, within that, we also have two types. We have what we call inferred databases, whereby the the data that is going there are inferred. They are not validated. They are not scientifically validated. There are no experimental backup for them. So those are inferred databases. These are databases that are generated using different algorithms. And you have made allusion to it when you are doing your lecture that, you know, in terms of the reliability of the various algorithms that are AI-based that we are using, how reliable they are. And there are also databases that are curated and also validated by experimental processes, by laboratory, by what's it called, experimental validation. And that is what's it called, the most reliable. And of course, most of this costs a lot of money. I mean, and by the time you are going to be assessing it, some of them are not free, because again, you have to pay the curators, you have to pay for the validation. A lot of this was being sponsored by NIH. You know, most of especially the genomic databases that I work with. But unfortunately now, you know, we all know what is going on in world politics, especially in the US. Funds are being cut, and the future of these databases are also threatened. So in terms of one person just sitting by in the corner of his room, no matter how well-intentioned he is, the veracity, validity, and the richness of that database will be very limited. So what I'm saying is that building a database is not just about only one person. This is multidisciplinary. The computer scientists have to be there because, hey, sorry, what are we talking about? All the algorithms that are working with PubChem, for example, is not an easy task. What's it called? Whether it's a biological database or a chemical database, the professionals have to be there. The curators have to be there. And these are also databases that are updated on daily basis. You will agree with me that when you are quoting your database in your publication, you have to state the time that you retrieve the data. Because again, they are being renewed every day. Data are being thrown out. new data are coming in. They are being renewed every day. So it's a dynamic thing, which is again one of the limitations of a book serving as a database. Thank you.
Wow.
So with that, I'm thinking that to get to have a kind of local database, or maybe Nigerian or West African database, a proper one like has been explained, I'm thinking that we can't just start from A. There are already these documentaries. So I was asking that, how do you verify? So we can just like when you want to do, no, let me not start with that. But instead of just starting from nothing, we already have documentaries to work with that can be verified. So it is that verification that should be like a next step. I mean, my own thinking, that's what I have to say.
Those documents contain some information, no doubt about it. Let's take, for example, protein database, the PDP. For people that have or Swiss protein or any of the databases, for those that have used some of these databases, the amount of information there cannot be contained in a book, no matter how well-intentioned you are. Yes, some of the information that's already in those books can be useful in building the database, but it is not enough. The amount of information there, what can be in that book? Classification, what's it called?
I said instead of starting from nothing.
Yeah, of course, they are going to be contributory.
Yes.
Contributory, yes.
And then those databases that are already established, I actually see that many of them have their authors. When you pick up, when you check a database and you want to use it, there are some places where they have uploaded the work and their authors, the people that worked on it. So those databases are actually the data there. They are from people's lab work, and the authors are written there. They will
be contributing. The point I'm making is that it is not limited to that alone. It's an all-encompassing thing that involves multiple professionals.
Of course.
It is the contribution of these multiple professionals that that determine the richness of that database. Because when you go to a database, you want to get the maximum information as much as possible.
Of course.
Especially if it's a validated, database.
I'm just saying that it's a starting point. I mean, it can be useful as a starting point instead of nothing.
Yeah. Yes. It can be. It can be. So, Dr. Chidi.
Wow. Yeah, I'm still with you.
It was a question that took us round and round, and we are coming back.
We are really, as if we drifted a bit, but it's still, very, very important we get this fact.
So we are saying that one in natural...
Just like you were talking about people who were given...
I was showing you...
develop, yes.
Yeah. Database and...
Hello.
What... So, yes, can you hear me?
Yeah, I can.
So, what I noticed was that they just did something and left it. Now, if you're into natural products, you can do research on database as little as you can. Then you can do research on, of course, in natural product now. But let me even put it to us that you cannot do much work on natural products without mining these databases. So it has an application in your research. Now, one of the applications where you take it to your research in natural products is this. Do my training, do my slide presentation. I never wanted to dip more into. I don't want it to be any career specific, but sorry. Now, if you are, let's say you're working on, you want to do a work on herbal formulation, which has been what we have been preaching since 1 month now. You need enough data. You need enough bioactive compounds to screen the potential of your plant. So where you are doing your own GCMS, and you are only having 50 compounds from your own GCMS result, you see that it is not enough to prove that that plant has the potential to treat the disease what you intend using it for. So until you create at least to an extent of 80% of that plant bioactive compounds. So for you to be able to create this, it means you will be using some database, some Chemoinformatics database or some pharmacology database. So if these databases are not there, it simply means that you'll be frustrated in your research work. You'll be totally frustrated in your research work because you need these databases to gather enough bioactive compounds that can make you draw conclusion that, okay, this I've gathered about 1,500 compounds from this plant, and so far a good number of this compound is pointing towards their anti-plasmodial activity. So it means that I can use this plant to control plasmodial disease because you have already gotten a large number of compounds. So you need chemoinformatics. So it is either you are building it or you are using it, but you need it in natural product study.
Okay, it's okay.
Yeah.
All right.
Thank you.
Yeah, thank you very much. Yeah.
All right.
Then, okay.
Yes.
Hello, Sir Henry.
Yeah, go
Yeah.
ahead.
What about applications like AdMets? Are they also part of Chemoinformatics?
Yeah, good.
Yeah.
AdMet is a chemo. In
Hello?
fact, AdMet is yeah. I'm coming. Let me attend to Chidin Joko. AdMet is
Yeah.
purely a chemoinformatics database because it does not have any business with biological data. AdMet does not work with, but any database that makes use of chemical data that retrieve information
Yeah.
relating to a chemical structure, a chemical to a molecule. Such a database is what we call the chemoinformatics database. But if you are retrieving, information as regards to a biological molecule, then that one is the bioinformatics. So AdMet is purely a chemoinformatics database because you are getting properties, PK properties of a compound, of a chemical molecule. So that is another application where you can bring in fact, the parameters we are having in AdMet, just like Professor Rotimi said, you keep updating it because the parameters we have before in AdMet is not what we're having now today. They keep increasing it. But you must know that a good database must be built on experimental backup. Despite we are using algorithm to push up to develop other parameters, but they must be built primarily first on the experimental backup for validation. They must be built primarily. If not, it's just as if you are building a database that does guesswork. That does guesswork. Yeah.
Okay. Yeah, thank you. Thank you very much. Yeah.
Doctor.
Before we take Daniel, okay. Daniel, Dr. Daniel, okay, you can go ahead.
Thank you very much, Dr. Henry. Thank you so much for the knowledge you are bringing to the table. I know that you are not a fan of publication. However, those within the academy is still part of our jurisdiction, so we cannot do without it. When a student goes and uses a particular plant, probably with available resources, the student uses 3 solvents and then works on a plant, does extraction and everything, then gets the GCMS results. Now, for that student to publish with just what the student has done, hence a publication.
Yes.
Now, from what you are teaching, what we are learning now, we have to go beyond that. That does not mean that this what the student has done should be negated, because assuming a supervisor has a lot of students and shares this into parts, I believe that the database or what we are trying to do now, if the data is generated from different sources and then collated along the line, or at the end of the day, and after some studies, say up to 10, you bring everything together and you do a work that could be a little bit larger, even if it's something that has to do with systematic review. You get something. But I believe that these other aspects should not be discouraged. That is my own thinking, because at the end of the day, these databases are things that are stored over time. They are not things that automatically. And people with time begin to subscribe and begin to use them. And so encourage us to go bit by bit, since resources might not be there to get everything done in larger forms, just like we are talking about big data now. Sometimes it is capital intensive. And so while we are encouraged to do this one too, I know you are not a fan of it, but we cannot do without it, sir.
Yes. You know why? Dr. Daniel, you is well. Well, why we are preaching what we are preaching now is because for many years, for years, we have been on the traditional aspect of our research work. We still do it because we cannot throw it away. Yes, we still do it. But while we are doing it, let's also look ahead. Let's try and look ahead. So I really appreciate your contribution. Of course, for now, your ranking depends on your number of publications. Do you know, Dr. Daniel, that I did a work for somebody, and what that person did was, this is a full work. What the person did was, okay, the work was published. Okay, that was a PhD work, yes, at IMSU, at the Imo State University, whoever, a PhD work. The person had to develop 8 topics, splitted this work into 8 pieces. Just now, she has published about 3, and Omo, the supervisor is really clapping for her. You are doing well. See papers you are generating. Me, I'm just looking at them. I say, well, which nonsense be this? But we will not stop. We'll keep doing it, because that is where we are find ourselves, where we rate scientists by paper. So we'll keep doing it. But while we are doing it, let's look a bit ahead into product, prototype, and chemotype development. So once again, thank you for that wonderful contribution, Dr. Reke. Yeah.
Hello,
okay, Salusi. Yeah. Salusi. Let me take Salusi. I saw your hand up before. Can go ahead. Is Salusi there? Imwana.
Yes, sir. Can you hear me? Yes, sir.
Oh, yeah. Go ahead.
Okay, sir. So, my first question is this: do you think in the future, there will be no free data accessibility. That is for now. If you can access PubChem and the protein database to download any data that you are interested in free of charge. Do you think in the future they will ask you to pay for a small person for you to access a particular data, since they are also investing huge amount of money to keep the database working? And the next question, sir, is this:
Yeah, go ahead.
okay, sir. So, initially, earlier today, I in the LinkedIn, I tagged you for a post about a one researcher from Nigeria that deposited an implicated protein for malaria type. So does that mean in Africa, or particularly in Nigeria, we can now perform a Nobel work, something like that work that I tagged your name in the LinkedIn?
Yes, let me come from the last question. I came across that. I saw that you tagged me, and I really love that because that is an improvement. That's an improvement using our local genomes. Now that is to say that people are doing novel work, and such a protein now we should have local database where those proteins can be deposited, stored. Because, like you cited, we should imagine that, just imagine that you are meant to subscribe to this already known database. You know this ideology when we are looking for to download journals, and then you just opened one splendid journal and you're asking to pay in euros to pay for it. What do? You will just keep it immediately and look for the ones that are open source to download. Because of that mindset, an average African will not like to pay to get something. An average African, because you just copy that title, the title of that journal. Maybe that publication was so useful. You copy it and begin to look for other data source that can give you open source for you to download. Now, that is to say that if this database, if they become locked that you subscribe, what happens to us in research? Because not everybody can pay such amount of money to access them. So that is a need for we now. And I'm taking it to our departmental level. We have data we have generated. At least we can start small. Start creating this data small. Just start doing something. Start with what we have. Data for us here. Can start doing something in our local countries. Yeah. So that is a powerful contribution from Esalusi. So because if this data,
so recently I noticed that whenever you are trying to access PubChem, they will tell you that due to the funding government, you may not find the up-to-date information about what you are looking. So what is the way forward also looking?
Of course, they are telling you that very soon they will ask you to pay to access PubChem. And if that be the case, a lot of us will be handicapped, those who love docking.
Dr. Henry, the issue with the PubChem has to do with what I mentioned earlier. You know, PubChem is hosted by NIH. And part of the focus of the present administration in the US is cutting funding for most of the international consortiums that the US was funding, you know, and that is what is happening to NIH. I believe that with time, they will get their bearings, they will get their footings back, maybe not now. Fortunately for now, they don't have funds to renovate or update the database, and also offer, some assistance. For example, if you're in a PubChem and you run into trouble, you can contact them, they can assist you. Those kind of supportive services are not there now because of, the withdrawal of funding for them. But I believe that with time, US is the greatest beneficiary of PubChem, whether you like it or not. So I believe that that is a temporary thing, and it will be resolved. But one thing I know is that whether we like it or not, science is the driving engine of humanity. And if we kill science, then humanity is going to go into extinction. So I don't believe that, as scientists, we will be shut out of databases. It's necessary for our existence as humanity, and it will continue to be there. So I don't think we need to be afraid of that. The only thing I want to say is to support you on the core for the building of our indigenous databases. For so many reasons. There are plants that are indigenous to us, that God has given us to explore, to solve our own peculiar problems. The Europeans, the Americans will not do that for us. It is our responsibility to do it, and to do it, we can. We have what it takes to do it. It's just for us to get ourselves organized and put things together. Thank you.
I am just a little note. I think Dr. Henry's apprehension is that we may be asked to pay. It will still be there, but it may not be at a cost, and that will not be too comfortable for us.
Yeah, but even now there are databases that are based on subscription.
No, he's not talking about those ones. He's talking about this one that we use generally.
PubChem. Well, I don't know. I don't work in PubChem, so I can't speak for them. But one thing, as I said, I'm sure is that, one thing that, nature abhors is vacuum. If PubChem goes, I'm sure something else will come that will be able to.
That will be.
But I'm sure that scientists will not be shut out of data. That I'm very sure.
Yes. So the call is indigenous database, very, very necessary. We might actually put up that call to see people who are interested to work with us. Already we are at Hendeb. That program we started it 2 years ago, and we are still working on it. More data no be here. We are still working on it, but we believe that we'll come up with something for Nigeria indigenous medicinal plants. Okay, I think I want us to have a little hands-on, but thank God for this interaction session. So it has opened us much knowledge. We shall now have the hands-on by tomorrow, 8 p.m. same time. I appreciate a participant from other countries who should be sleeping by now, but they are still awake. I don't know what they are doing, awake listening to Chemoinformatics. It's actually great. Yes, that is part of the price you pay. Just the same way too at Hendeb, we have not had rest. We only rest on Sundays. Yes, we only rest on Sundays. But even that Sunday rest is not even a rest, because you go to church, you come back, you do other activities. But we are still pushing it, because we are aiming at a point. We are aiming at achieving something. So we say thank you for your effort. The video will be released. Our materials, just as you know, that we are expected to make a little token of 2,000 naira for the local participants, and 3 USD for our international participants. So we can grant you access to our materials and our videos. One thing that we have done this year is that to all our training, we are giving you supportive textbooks. But I know that we are not reading them, but just have them with you. One day you need to go back to them because of the time. Yes, just have them with you. They will help you to have a theoretical knowledge of what we are discussing. So we have about 4 to 5 textbooks on chemoinformatics. So it will help you and give you good knowledge on what we are talking about. So by tomorrow before 10 a.m., we shall be giving access to those who have made that payment. So if you still want to make the payment, you can still go ahead. We sent out a recent mail, but we noticed the former mail we sent had some contradiction to it. So we sent a recent mail today, which I know we have all received that. Some of us are not also in the WhatsApp platform. I would appreciate you join the WhatsApp platform for further informations. So if you're not in the WhatsApp platform, you can chat us up with the number in the flyer. Just send us a DM, so we'll add it up to the WhatsApp platform. So once again, we want to say thank you. I appreciate all our contributors who has contributed in one way or the other in this training. We are going somewhere, and I believe that we are going to get there. Thank you once again.
Before you go,
So
Dr. Henry, some
Yes,
of us didn't receive mail today.
Okay.
Help us,
Okay.
Yeah, help us to look into that. Some of us that paid did not receive the mail.
But you mean some that registered did not receive mail?
I registered, but I didn't get any mail.
Okay. We'll get that notified. That means those that, and that was the reason why I had to send the link to the platform so that we can resolve that mail. So those who have not received the mail, you just drop your email with us in the platform. Drop your email. So we are going to definitely before I take a rest. So before I take my rest, I'm going to reconcile that email. We are going to do that with our technical team. So once again, thank you for that observation. We are grateful having you. Some of you have been joining in all our training. I appreciate you. I really appreciate you for that energy. Thank you so much. By next week, the flyer is not yet out, but it will be out tomorrow. We shall be having a webinar discussion by next week. And it will be so awesome. So we'll have time to discuss on issues surrounding in silico studies in Nigeria and Africa at large. So good night to every one of you. We'll meet tomorrow.
Thank you, sir.
Thank you.
Good night, everybody.
Yeah. Thank you.
Good night, sir. Have a nice night rest.
Yeah. Some of you that your time now is morning. Good morning.
All right, thank you.
Hello, sir.
Thank you.
Yes. Andrew.
Sir.
Yeah.
Sir.
Yeah,
Hello,
go
sir.
ahead. I can hear you.
Sir, I don't know if it is right to say this thing in this time, but I have been forgetting to say it, sir.
Okay.
Sir, it is about our previous training on computational chemistry. You made mention that we are going to discuss about how to generate this structural data from Hendeb. So it was after we finished that session, the thing come to my mind again. So I keep on forgetting it to remind me. So it is now that I now remember I say, let me say it out so that I should not forget it again. I know all the people participants here could remember about what we said earlier.
Yeah.
So we are going to talk about.
Yeah, you keep me on a reminder now. What we'll do is on the day of our defense for that beginner's course, I will show us how to generate those spectral data from ChemDraw.
Okay.
But you can try it to, yeah, but I would, on the defense, where we meet again, then I will then show us that it's just a little procedure.
Okay.
Or I can still send the process to the platform, then you try it.
Okay. That's
Yeah.
right.
Okay, thank you.
Yeah, thank you. Thank you. We are really improving, and I like that. Thank you. Good night, everybody.